Hello, this is Yashveen. In this video, I'll be solving paper 23, 9700 Biology, May-June 2020. Starting with question number 1, water and mineral ions are transported up the stem of a plant to the leaves within xylem vessels. Some water and minerals can pass out of xylem vessel elements to supply parenchyma tissue in the stem. So here you have a plant diagram of a section through a stem. Identify one location where xylem vessels occur in the stem by drawing a label line and the letter X on figure 1.1. So the label line should be pointing to the inner side of any vascular bundle, which would be xylem. B. Explain how hydrogen bonding between water molecules contributes to the movement of water within xylem vessels up the stem to the leaves. Here, the movement of water is due to transpiration pull in the stem, which includes continuous unbroken columns of water aided by cohesion, which is the attraction between water molecules due to hydrogen bonding. See figure 1.2 is a diagram of a photomicrograph showing 3 adjacent parenchyma cells in the stem. The parenchyma cells can be described as typical plant cells, so they would have all the structures within a plant cell. The arrows show the diagram of movement of water between the cells. 1. Describe and explain the movement of water shown in figure 1.2. The movement of water occurs by osmosis due to a difference in water potential gradient, and that means the movement of water over here is going to be down the water potential gradient by osmosis, but as the question is of 3 marks, you'd need to elaborate. And as shown by the arrows over here, the route is a symplast pathway as it crosses through plasmodesmata and travels through the cytoplasm of the plant cells. 2. Only some of the structures visible using the light microscope have been included in figure 1.2. List the features that can be seen using the high power of a light microscope that can help identify a parenchyma cell as a plant cell and not as an animal cell. So any features of a plant cell that distinguish it from an animal cell, which would include a cell wall, plasmodesmata, and a large vacuole in the center of the cell. In 2016, the highest number of cases of malaria and deaths caused by the disease were in sub-Saharan Africa. In many areas of sub-Saharan Africa, malaria is an endemic, meaning it is continually present, and people are at high risk of becoming infected with the Plasmodium pathogen. In high-risk areas, it is recommended that homes are provided with insecticide-treated nets, short ITN for short, and the surfaces inside homes where Anopheles mosquitoes may rest are sprayed with insecticide. This is known as indoor residual spraying, or IRS for short. A. Explain how the use of ITN and IRS can help break the transmission cycle of malaria. The transmission cycle of malaria involves mosquitoes acting as vectors, so they need to take blood meal from an infected person to transfer the pathogen from that blood meal to an uninfected person, and this is part of the transmission cycle. Using ITN and IRS, it would be ensured that the Anopheles mosquito is killed either before taking blood meal from an infected person or after it has taken blood meal from an infected person, but before it has taken second blood meal from an uninfected person. This way, it would be ensured that the Plasmodium pathogen is not transferred from an infected person to an uninfected person, therefore breaking the transmission cycle of malaria. B. Figure 2.1 shows the proportion of population in sub-Saharan Africa at risk of malaria that is protected using IRS or ITN or both in the years 2010 to 2016. So here's the bar chart, and the main trend in figure 2.1 shows that there is an increase in the percentage of the population protected over time. One other trend shown in figure 2.1. So you can see that the darkest shaded area of the graph is increasing, whereas the area shaded by gray color is decreasing. So that could be a trend you could write in your answer. which is the percentage of population protected by IRS only. On the other hand, you could also write how the percentage of people protected by ITN only is increasing with time, which is the darkest shaded area of the bars. To explain why the main trend shown in figure 2.1 could be a concern for World Health Organization. If the number of population protected by IRS is decreasing, that could mean the mosquitoes are becoming resistant to insecticide which is used in the IRS, meaning that IRS is slowly becoming ineffective. 2. With reference to figure 2.1 suggests a reason for the difference in trends shown for ITN only compared with IRS only. So you can see in the trends that ITN only treatment is protection by ITN only is increasing, whereas protection by IRS is decreasing. Since both are using insecticides, that could mean that the insecticide used in ITN is more effective in killing mosquitoes. C. In a primary immune response, antibodies against Plasmodium are produced within 1 to 2 weeks following infection. In some people, the pathogen is eliminated and the concentration of antibodies in the circulation decreases over time. Infection against Plasmodium with the same antigens causes a secondary response that also involves antibody protection. State and explain how the antibody response following a second infection will differ from primary immune response. Antibodies production in a secondary response is faster and creates a greater concentration of antibodies than primary response, and this happens because memory cells which had been produced during the primary response are now present in the body, and they are able to form many B lymphocytes that secrete antibodies. In malaria, the production of antibodies is beneficial to recovery, whereas in the disease Myasthenia gravis, the production of antibodies is harmful. Explain why the production of antibodies in a person with Myasthenia gravis is harmful. This disease is an autoimmune disease, meaning that antibodies attack self-antigens, so they work against self-antigens, which is why their production is harmful. 3a. Figure 3.1 is a photomicrograph of a section through two different types of blood vessels, X and Y. One, name the two types of blood vessels shown by X and Y in figure 3.1. As you can see, the diameter of lumen in X is smaller relative to the thickness of the wall, meaning that this is an artery, and also the tunica intima has cripply edges, unlike Y, where the tunica intima is smooth and the lumen diameter is wider relative to the thickness of the wall. So X is an artery and Y is a vein. To state the reasons for identification of the type of blood vessels shown by Y in figure 3.1. So again, Y has a smooth tunica intima and the size of lumen is large relative to thickness of the wall. B. Tissue fluid and lymph are formed when blood arrives in the capillary networks of body tissues. 1. Explain why tissue fluid is more similar to blood plasma than it is to blood. So, blood plasma on its own and tissue fluid don't contain red blood cells, unlike blood, because red blood cells are too large to pass out of the gaps between endothelial cells of the capillaries, and so they can't leak out of the blood with plasma to form tissue fluid. To explain why tissue fluid formed after blood arrives in capillary network has a high concentration of amino acids than the newly formed lymph draining away from the network. So lymph forms after tissue fluid that forms from the blood has passed between the cells, and during the tissue fluid flowing between these cells, the cells take up any required amino acids, and so in the time that tissue fluid forms lymph or drains into lymphatic vessels, all of the amino acids have been used up. See, the trachea, bronchi, and bronchioles in the gas exchange system require a supply of glucose and oxygen from the blood for the functioning of smooth muscle. Outline the function of smooth muscle in the gas exchange system. The muscles contract and relax to change diameter of all of trachea, bronchus, and Saccharomyces cerevisiae is a unicellular fungus that is important in the brewing and baking industries. Figure 4.1 is a diagram of transmission electron micrograph of S. cerevisiae. Where you have glycogen granules, cell wall, cell surface membrane, lysosome, vacuole, lipid droplet, and a nucleus as well. A student was asked to calculate the magnification of the image shown in figure 4.1. The student began by measuring the length of the scale bar in millimeters using a millimeter rule. State what the student should do next to obtain the correct answer. His next step would be to convert his readings in millimeters to micrometers by multiplying it with 1000 or 10 to the power of 3. The one function of the liquid droplets shown in figure 4.1 is to store triglycerides. The triglycerides in a lipid droplet are surrounded by a single layer or monolayer of phospholipids. Suggest and explain why phospholipids rather than triglycerides are used for the outer monolayer of lipid droplet. The outer monolayer interacts with the aqueous environment, which is possible for phospholipids due to their hydrophilic phosphate head, whereas triglycerides are hydrophobic, therefore they cannot interact with the aqueous environment, and so they cannot form the outer monolayer. See, the lysosomes and vacuole of S. cerevisiae contain acid hydrolysis or hydrolytic enzymes that function in an acid pH. Explain why lysosomes need hydrolysis to carry out their function. Hydrolysis carry out hydrolysis, which is the breakdown of a substance by addition of water, and for a unicellular fungus, that could be breaking down worn out organelles, as these organelles would be no longer of use. A disaccharide trehalose is a reserve store of energy for S. cerevisiae. When glycogen stores decrease, the monomer of glycogen and trehalose is an alpha glucose molecule. 1. Complete figure 4.2 to show the ring structure of one alpha glucose molecule. So it's supposed to have the hydroxide group downside and a hydrogen similarly, whereas on the third carbon atom, the hydroxide group is projected into the ring. And here you need to add the OH, and that is it. 2. A student carried out tests on a solution of trehalose and correctly concluded that trehalose is a non-reducing sugar. Outline the procedure carried out by the student and state the results that were obtained. To test for a non-reducing sugar, you'd first have to heat trehalose, in this case its solution, with Benedict's solution, which would give a negative result since Benedict's solution tests for presence of reducing sugar. Then you'd have to boil the trehalose solution with hydrochloric acid and then neutralize it with, for example, sodium hydroxide, which is an alkali, after the solution cools down. And then you'd have to repeat the Benedict's test, which would now give positive results, as the non-reducing sugar would be broken down to reducing sugar. And depending on the concentration of trehalose, the color could be green, yellow, orange, or red. The hydrolysis of trehalose is catalyzed by two different enzymes produced by S. cerevisiae, regulatory trehalase and non-regulatory trehalase. A study was carried out to compare regulatory trehalase and non-regulatory enzyme extracted from the species. The results of the study showed that regulatory enzyme had a higher Michaelis-Menten constant value than the non-regulatory enzyme. The optimum pH of the regulatory trehalase was between 7 to 7.8 pH, whereas the optimum pH of non-regulatory trehalase was somewhere between 4.5 and 5. 1. Explain what is meant by a higher Michaelis-Menten constant value. Since Michaelis-Menten constant is the substrate concentration required to reach half of the maximum enzyme velocity, it means that high Michaelis-Menten constant means that a higher substrate concentration is reached for enzymes to reach maximum velocity or work at their fastest pace, and that means the particular enzyme has a lower affinity for its substrate. So, lower enzyme affinity would mean that due to less attraction between enzyme and substrate, it would mean that the enzyme substrate complex formation is not easy, and therefore a high substrate concentration is required for all of the active sites of the enzyme to be occupied. 2 regulatory tree helase is found only in the cytosol, which is the fluid part of the cytoplasm, whereas non-regulatory tree helase has been found on the external surface of the cell surface membrane and inside the cell. State the location inside the cell where non-regulatory tree helase is likely to be found and explain the reason for your answer. So non-regulatory tree helase enzyme has an optimum pH of somewhere between 4.5 to 5, which is an acidic pH. Lysosome and vacuole have acidic environment, and therefore this enzyme would be found in 3. Explain why both types of tree helase, regulatory and non-regulatory enzyme, can be described as intracellular enzymes. That is so because both of these enzymes can perform inside the cell, so they'd be called intracellular enzymes. F. Saccharomyces boulardii is a strain of S. cerevisiae. It has been researched for its possible health benefits for some gut diseases. Researchers investigated tree helase extracted from S. boulardii, concluded that only one type of tree helase was present in the extract. Figure 4.3 shows the effect of pH on the activity of tree helase extracted from the second species. With reference to figure 4.3 and to two different types of tree helase enzyme produced from the first species, state and explain what can be deduced about the type of tree helase present in the second species. The two types of enzymes mentioned in the question are regulatory and non-regulatory. The enzyme in the figure works best at around 6.7, which is closer to the first range, 7 to 7.8 pH, then the second range, which is 4.5 to 5. So it is more likely to be a regulatory tree helase than a non-regulatory tree helase, as its activity peaks at a pH closer to the optimum pH range of regulatory tree helase. 5 blood cells are formed from tissue stem cells in the bone marrow. These bone marrow stem cells go through a number of mitotic cell cycles to form the fully functioning blood cell. Figure 5.1 shows 3 main types of stages of cell cycle. The activity of genes during the mitotic cell cycle. When genes are being expressed, the cell produces many messenger RNA molecules and ATP molecules. Explain what is meant by the term gene. Gene is a length of DNA which codes for polypeptide. So the nucleotides in the length of DNA that would code for mRNA during transcription, which would then be translated into the length of polypeptide. B. The main stage of the cell cycle, during which most mRNA and ATP is formed. It is the interphase, as interphase has the G phase, where cells grow using proteins that are made using mRNA and ATP molecules. See figure 5.2 is an incomplete diagram of an ATP molecule. One on figure 5.2 complete the diagram of ATP molecule. So ATP stands for adenosine triphosphate. So you'd need 2 more phosphate molecules to complete the 3. And write the name of the base in the space provided. This would be adenine, and the name of the sugar is ribose. 2. The base shown in figure 5.2 has a double ring structure. State the name for a base that has a double ring structure. That would be a purine base, since pyrimidines contain a single ring. These suggest and explain the role of mitosis in the formation of blood cells by the bone marrow stem cells. So stem cells are present in bone marrow, which would form red blood cells by mitosis, and you need mitosis because without mitosis there would be very few red blood cells. So you need mitosis to produce a large number of genetically identical red blood cells, and these are required for oxygen transport. 6. A student carried out an investigation to estimate the water potential of potato tissue. The main steps in the procedure and in the analysis of results are outlined in figure 6.1. So in the first step, 6 different concentrations of sucrose solutions were prepared and an equal amount of each was placed in a labeled beaker. So you have 6 beakers with different concentrations of sucrose. 6 equal-sized blocks of potato tissue were cut out of the same potato, blotted dry, and weighed. One potato block was immersed in the solution in each beaker for 30 minutes, so each different concentration solution had a piece of potato tissue. After this time, the block was removed, blotted dry, and reweighed. The experiment was repeated twice. The mean percentage change in mass of potato tissue was calculated for each concentration of sucrose. A graph was drawn of mean percentage change in the mass of potato tissue against concentration of sucrose. Explain why different concentrations of sucrose result in a different mean percentage changes in mass of potato tissue. Water moves down the water potential gradient by osmosis, and this change in mass occurs due to movement of water either into the potato tissue or out of it, and the movement depends on the concentration of the beaker it was put into and the concentration within the potato tissue. Water moves out of the potato tissue and into the sucrose solution if the solution has a higher sucrose concentration than potato, and therefore the mass of potato decreases, whereas water would move into the potato tissue if the solution of sucrose has a lower concentration of sucrose or a higher water potential than a potato. Therefore, the mass of potato would increase. B. State how the graph is used to estimate the water potential of potato tissue. Okay, the concentration of sucrose solution that corresponds to no change in mass of potato tissue will be the concentration of sucrose in potato, because net movement of water will be zero at this point, meaning that the water potential gradient does not remain, and that would be used to find potatoes water potential. And this is it, we are done with this paper. Thank you for watching.